This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 328, recorded on March 13th, 2015. And it's Friday, so it's Friday the 13th. Hey everybody, I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today, you can tell by that heavy breathing, Dickson.
Oh, stop it.
Despommier.
If I didn't breathe heavily, I wouldn't be breathing at all. Hello, Vincent.
You know, when I edit, I play the beginning, and I hear you breathing all the time.
I can't help it if I breathe in and out. I'm a mammal.
I thought you were a camel.
No, otherwise I'd be producing nanobodies.
Ooh, you actually listened.
I listen. I definitely listen.
I'm waiting for you to get mad at me.
I'm not going to get mad at you, Vincent.
The other day we did a paper, and you started talking about the assay, and I said, Dickson, you actually know the assay.
Yeah,
you got so mad at me.
I resemble that remark because you have no idea of what I read unless you ask me.
No, I assume you read nothing. The default assumption. Also joining us today from southeastern Michigan, Kathy Spindler.
Hi, everybody.
Where you just got back from Indiana, right?
Yep, this morning.
All the way from Indiana?
Is that like a mile away?
I flew, I drove, and anyway, I'm here.
You drove some? Yeah?
Well, I had to drive to Indianapolis Airport, and then I had to drive home from the Detroit Airport.
Oh.
Yeah.
Well, welcome back.
Thanks.
Do you have nice weather?
Yeah, it was nice.
Good.
No rain, and yeah.
Had you been to West Lafayette before?
For 4 years for my undergraduate degree.
I forgot.
And many times subsequently, yes.
I've never been there.
Really?
I went to Notre Dame and I never got there.
Probably never go.
I never got there.
Probably will never go.
Well.
Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
How have you been?
Oh, doing okay. It's been above freezing just about every day this week, so I'm feeling pretty good about that. We do still have snow on the ground, though.
Yeah.
Is it black?
All along the roadways, it's all black. Mount Snowplow is gray to black. They've got the snow piles at the supermarket that are piled up from the parking lot that are still 15 feet, 20 feet high, and those are all black. But it's gradually melting.
And also joining us today from North Central Florida, Rich Condit.
Hi, everybody.
Hey,
How are you doing?
Hello, Rich.
You've been home for like 2 weeks now.
Oh, yeah.
Feel good?
I'm really locked in here, and I'm going to be home for a while, yeah. I got very little, if any, traveling on my calendar. This is good. And you need to know that it's 83 degrees Fahrenheit here, which is 28 Celsius. It is, however, cloudy, so,
Yeah.
But it's okay. I am wearing short sleeves and shorts today, so...
Oh, my gosh.
So, Rich, you're going to see us fly over you tonight because my wife and I are going to Miami to attend a wedding.
Excellent. Well, have a wonderful time.
It's even warmer down there.
Yeah, it's going to be warm all weekend.
I know it's not actually over, Richard, but it's close enough.
Oh, yeah. I mean, we got azaleas blooming and all that kind of stuff.
That's nice.
You're not actually going to see him.
No. We'll pretend we see
him. He's not even going to think of you.
This is true.
In fact...
He might. I might think of him, Might happen.
You see, there is some camaraderie.
Actually, my wife found some article that mentioned you about vertical farming the other day.
Really?
Came running in to show it to me. Yeah.
See, it's true. It's true.
Yeah.
I can't get the weather here. It's just...
But it's not so bad out. You can look out the window.
The temperature. I don't know.
It's about
39 Fahrenheit here, so it's probably pretty close.
That's
to that
about 42, 43 degrees.
What is that in Celsius? About 8?
I was just
4 Celsius.
Good.
So it's probably like 5, 6 where you are.
I'm used to Celsius. Today's Friday the 13th.
Yes.
Oh, that's right.
Everybody okay?
Yeah.
Yeah,
so
far. So good.
Yeah.
I got the recorder on. Let's see, because that would be a thing I wouldn't do, right? Backup is on. Dickson's here. All right, so this should work. We have a bunch of follow-up. The first one is from Robin, who writes, "Gorillas are people. Dear TWiV gorillas." Quote, "Chimps and gorillas are sympatric," unquote. We're going to get into that later. Consider politicians, lobbyists, economists, banksters, and lawyers as a group. Consider gorillas as another group. Towards which group do you have better feelings?
It's no contest.
Yeah, I do like gorillas better than those guys.
Here.
Next, they should study dung beetles or their equivalent with respect to gorilla dung to see what role, if any, they may have with the immunodeficiency viruses.
That's a very good idea.
On a completely different note, Scottish deer are culprits in bird killings. Apparently, deer are killing birds.
Huh.
Did you know that deer would do that, Dickson?
I had no idea.
Red deer on the Scottish island of Rum may be eating the heads and legs of live seabird chicks as a way to get minerals they need to grow their antlers.
I'll be darned.
Puts a whole new spin on Bambi.
I'll say.
Bambi took a beating last time. It's getting worse.
And he sent another link to an article. Field cameras catch deer eating birds. Wait, why do deer eat birds? And he also sent a photo, which I didn't put here, of a squirrel eating a bird.
Yeah.
Did you know that?
I once said, and I was right, that there's no such thing as a strict herbivore. I think if the meat is available, things will take bites out of them.
Well, what is this bird eating business? What did we say last time? That
we were talking about whether gorillas would ever eat meat in order to acquire
That's right.
SIV. And then we got into a conversation about animals that are supposed to be herbivores eating meat, and that got Robin rolling. Okay.
Gosh, I forgot all that.
Yeah.
What I like is this second article has a concluding line that says the famous line from The Simpsons might be accurate. If cows ever got the chance, if a cow ever got the chance, he'd eat you and everything you ever everyone you ever cared about.
Right. Speaking of The Simpsons, the guy who helped create it just passed away.
Yes. Right.
Dixon, you read too much Yahoo News.
That's all I read.
That's all you read.
So he's retired. He can do anything he wants.
I come in
thank
here.
you, Richard.
Well,
thank
Yahoo
You're
News
welcome.
is
I'm
actually
practicing.
useful
No, wait.
if you want to be able to have conversations with ordinary people.
That's true. That is true.
It serves the same function for me now that that glimpses of the New York Post on the Staten Island Ferry used to serve when I was living in New York.
That's
Wait a
right.
minute. I'm an ordinary person. I don't read
You
Yahoo
can't be an
News.
ordinary person. You're a virologist.
No, I'm an ordinary person. I'm
No,
just
no.
like everyone else
not.
here. Yeah.
But if
Do
you
you
want to
read-
know what everybody else is talking about,
Do you read the
or might be
headlines
talking
on the
about,
newspapers when you're standing in line to check out at the
No,
supermarket?
I don't read any headlines. No, not at all. I get the Times on the weekends, and that's it. And I'm thinking of canceling that because I'm mad at the Times.
Oh.
What'd they do to you?
Yeah,
So you know what's funny? Last Sunday, I get the magazine section, and I start fuming because there's usually no science in it. And I think every one of those articles should be about science. They got articles about fashion and watches and all this stuff that doesn't matter, except that it makes a lot of money.
Yeah.
And I open it, and there's a Carl Zimmer article. So I felt badly. But I do think they should have at least one science article every Sunday. Make
Well, they need you to write it.
Times had a big science article on the front page today.
About what?
Water distribution throughout our solar system.
Okay.
Apparently it's more common than we thought.
Now let's do some more follow-up. And I'm going to have Kathy take the next one.
Okay.
Right.
Trudy writes, Dear Twivers, the German word for shit seeker is Scheissesucher.
There you go.
Very well
If you're
pronounced.
ever in need of real-time translation from English to German or German to English while twiving, please feel free to text me. I also wanted to mention that I agree with your listener who asked for an episode on respiratory syncytial virus. While the lack of progress in this area, at least as far as a vaccine is concerned, has been frustrating, it might be worth updating listeners about where we stand right now and also provide some information about why the development of an effective RSV vaccine has been such a challenge. RSV infection is, after all, of notable clinical significance, especially for parents of infants and toddlers.
Right.
Kind regards, Trudy.
I feel like texting her something right now. What do we want to know?
Was bedeutet alles das?
What's that mean?
What does all that mean?
Really?
Yeah,
Do you speak German
a
too?
little bit.
Kathy, why don't you take the next one?
Oh, well, I was just going to address the RSV
Okay.
question. My former student, Marty, had an MBio paper last September And he also wrote a pathogens pearls for us, so he could do some kind of state of the art on RSV vaccines or something if we want to have somebody like him on.
That'd be good.
We should do RSV for sure.
Yeah.
He said there's a lot going on right now in the field. So there's a NIH fusion protein structure and why RSV is a special case and so forth.
Cool.
Yep.
Right.
Do the next German one, Kathy.
Well, Rich can do it too. He's had German.
You have
Give
German,
Kathy
Rich?
all the crap.
Yeah, I got a slug.
All right, Rich, go ahead.
Dear TWiV team, seeking for linguistic truth. Here's a small assortment of possibilities. Very technical. Excrementensucher. So that's excrement seeker. Literal Scheisssucher. Scheisse is very rude, but nobody would understand Scheisssucher. It, moreover, sounds like an insult similar to
Remember,
F-bomb
Caleb
seeker.
is listening.
Okay. And by the way, look at 3 consecutive S's. Yeah, in Scheisssucher. All correct. I told you the Germans are champions at what they call Wortbildung, building words out of smaller words.
Yeah, freight train sentences are good.
Kottzucker, where Kott is feces. 4, best choice, Kott-Sammler. Instead of seeker here, collector equals Sammler is used, but please not in the sense of collecting stamps. And it could be mistaken with the following gear for dog poop, and he sends a
show illustration.
Yes, a picture of somebody with some fancy device in a bag for picking up and bagging your dog poop.
Right.
And says, you see, Germany is very complicated. As always, many greetings from Wiesbaden, Germany, Peter.
Oh, that's a nice place.
And a little at Kathy. To find equals finden has the meaning of to discover. But I agree with you, Kathy. I don't remember that finden ever came up. Nevertheless. There you go.
4 different ways to say shit seeker.
Yes.
This is wonderful.
This reminds me of that little gag in the movie Top Secret. Oh, I know a little German. He's over there.
Yeah, who else can pronounce German?
I can pronounce it.
German. There, I just did.
Right.
All right,
I was
go
just
ahead,
going to say that.
Dixon, read Matthias's.
Matthias writes, now that the weather is bike-friendly once again, I get to listen to more TWiV shows on my way to and from work. On TWiV 327, you wondered about the German translation of shit seekers. Kathy's right with Zucker for seeker, or Scheisse-Zucker, would be the literal translation. However, a word of caution, do not omit the word, shucks.
Come on, Dickson.
Oh, Scheisse. Do not omit the E. A Scheisse-Zucker quickly transforms a diligent feces scout into someone who is doing a shitty job of seeking. Or, even worse, into an insult in terms of, oh, here comes another bloody seeker. If I had to find an informal German expression for these people, I would probably call them Kackersammler, crap collectors. But I'll stop here with this highly interesting scientific discussion. Matthias.
Except that I think you would pronounce his name Matthias.
Matthias, Dixon.
Matthias,
Come on,
Matthias.
Dixon. You said you could pronounce
You
German.
say potato, I say
So
stone door.
Matthias is a virologist in Germany who discovered virophages, working with Curtis Suttle over in Vancouver. He ran the giant virus meeting last year. 2 years ago, and I know him. He invited me to that, so cheers, Matthias. We'll say Matthias again to make up for Dickson.
Okay.
Alan, you want to take Steve's?
Sure. Steve writes, hi, Vincent. Congratulations on reaching your 100th TWiM, by the way. I don't always understand it, but I'm sure it will begin to sink in if you keep it going long enough. On the most recent TWiV, I have to say that I was quite impressed by your inability to find a more suitable word than people to describe our increasingly less numerous relatives. They might not always be the best mannered of people, but there are plenty of humans who are much worse. I have actually come across the use of the word person in zoological publications before, used for much lower life forms than gorillas. Lower in quotes. I cannot give you a reference, but I used to buy old science books that I came across in second-hand shops, and in an old marine biology book, I found that the individual polyps of the Portuguese man-o-war were called persons. I presume this was to recognize that they were individual members of a communal organism rather than cells of one organism. If in times gone by the individual personhood of the lowly polyp was recognized, there should be no problem with recognizing communities of higher apes as the people they, no doubt, think themselves to be. Keep it up.
Yeah.
What's the Y for?
I don't know.
Maybe that's a emoticon translation
It might
error.
be. Yeah.
Oh, it might be.
I thought when you type these things into Google Docs, it automatically does it. Let's try.
But you know how the smiley face comes out a J?
Yeah, it didn't work.
Yeah.
Whatever.
All right.
So yeah, in the historical scientific literature, there's often a lot of anthropomorphization.
Yeah.
It's interesting the things that garner follow-up, isn't it?
Yes, it is.
Yeah.
Means they're really listening.
Yeah, well, some things are just points, and others,
Yep.
But I appreciate it. That's great.
Nice.
Well, I guess Scheisse was just a fertile topic for discussion.
Very
Arg.
clearly. I did record Beatrice Hahn saying that, and I have to dig it up and we'll play it sometime because everybody laughed, of course. I love it. All right, before we go on to our paper for today, I just want to remind you that the ASM Press has published a book on microbial diversity. In fact, it's called Principles of Microbial Diversity. It's like Principles of Virology.
Oof.
Yes.
This would be a good textbook for upper-level undergraduates majoring in microbiology, genetics, biology, and it gives you a solid curriculum to explore the big range of biological diversity in the microbial world. It's written by Professor James W. Brown and his band of renown. No, that's someone else. So they're never going to take ads out again, are
That's
they?
an old reference, too. I'm surprised.
James Brown and his band of renown?
Well, the band of renowned phrase, it's hearkening back to the 40s and the swing band
Yeah.
era.
Well, I thought you would get that.
I like it.
I think that it's good because it gives people a mnemonic to remember the author's name.
Here.
Yes.
Anyway, James Brown has taught microbial diversity for over 20 years at North Carolina State University.
Great school.
And this offers a survey of the tree of life that establishes what you need to know about the microbial world. It does fill a niche in microbiology textbooks. Ha, ha, ha. Get it, Dickson? A niche.
Yes.
I got it.
By providing an engaging, cutting-edge view of microbial diversity. You can find it at asmscience.org. It's a website you go to buy it. It's $90. Check it out. You can go look at it.
Worth every penny.
All right. We have a paper that caught my eye today. And my eye is hurting me at the moment, so pardon me while I scratch it. Okay. This paper seems to have gone right into my eye, Dickson.
Uh-oh, because it has a reference to eyes later, too.
It does?
Yeah.
How so?
The retina pigmentosa.
Ah, you're right.
Oh, yes.
Wow, you are something, Dickson.
See,
We ought to put you on a podcast.
No, nobody would listen.
This paper's about picornaviruses,
Ah.
and I would not normally pick a picornavirus because of that, but I found it really interesting.
Isn't polio a picornavirus?
Yeah, it is.
That's why I thought you picked it.
Although it's not exactly about polio, but it's very
Okay.
related.
All right.
It's published in PLOS Pathogens, and it is called Cytoplasmic Viral Dna, RNA-dependent RNA Polymerase, disrupts the intracellular splicing machinery by entering the nucleus and interfering with PRP8. The authors are Liu Kuo Lin Huang Liu Arnold Chen Wang Cameron and Shi. And they are from Taiwan and Penn State University. Craig Cameron is a well-known enzymologist who studies polio RNA polymerase. And this is all about the RNA polymerase of these viruses. Now, as you know, the RNA polymerase replicates the RNA genome of picornaviruses. They all have one. RNA-dependent RNA polymerase. And an interesting finding is that this protein, so these viruses are cytoplasmic. They replicate in the cytoplasm. In fact, if you take the nucleus out of a cell, polio is happy to replicate in it. It ignores the nucleus. I guess.
Do we need to mention that these are naked single-strand RNA viruses?
Yeah,
or
absolutely.
everybody already knew that.
They're
do need
naked.
to.
They're absolutely naked.
They come in naked and they go out naked. Yep.
That's right.
Except Hepatitis A virus
Right.
sometimes acquires a membrane.
I think that's going to become more and more common.
And I understand
Now that
that
people are looking.
I understand that someone has looked and found the same with poliovirus.
Yeah, I heard the same thing.
So it's very interesting. So they're only naked sometimes. If they're not in the right home, they're naked, which is like Hep A.
Naked and afraid.
If you don't grow it in a liver cell, it's naked.
Yeah.
Anyway, so they don't care about the nucleus, sort of like pox viruses, right, Rich?
Right. Well, pox viruses kind of sort of care about the nucleus.
If
you inactivate the nucleus, they kind of die late during infection.
Yeah.
They can't make it all the way through.
See, I have to.
And
I
that's
have to
not
point
understood
something
why.
out here. You accuse me of not reading anything. I once brought an article into you, Dr. Racaniello, called "Kickstarting a Viral RNA Polymerase," and you thought it was a good article, but then we didn't do it, and it relates to what we're doing today, doesn't it?
Maybe we will do it.
Maybe we will. That's right.
Yeah, it's a Hep C. It's a very interesting article on the Hep C RNA polymerase.
Yeah.
So these viruses like to replicate in the cytoplasm, but some of their proteins go in the nucleus. And so the polymerase is one of them that does this. The polymerase is made as a precursor with the preceding protein. So these viruses have an RNA genome. It's a plus-stranded RNA that is translated into a long protein, a polyprotein, then it's chopped up by viral proteases. So the polymerase, which is the last protein in the genome to be translated, is made as a precursor with the protein before it, which happens to be a protease. All right.
And then the precursor goes into the nucleus, right?
The precursor goes in the nucleus because there's a nuclear localization signal in the polymerase, and the protease takes a ride.
How do they do these? Come on, you couldn't make this up.
Where is the nuclear localization sequence? Is it in the 3D
it's in
part?
The 3D. It's in the 3D part, right?
That's the polymerase.
Okay.
And then the reason the protease gets in is because then it inactivates transcription factors.
So where
nucleus.
does that signal peptide actually originate? I mean, who gave it to that virus?
What? The nuclear localization signal?
Yeah, exactly.
Who gave it to it?
Yeah, I mean,
no one gave it to it.
Where did it pick
It
that
just
up?
took it.
Just
I mean,
evolved.
that's incredible.
It fulfilled the need.
You think about these safe cracking episodes that viruses go through in order to crack the
Safe
code.
cracking. I do like that word.
This is one of those most remarkable events that allows it to do both things at the same time. The cytoplasm and nucleus. I'm astounded.
It evolved at some point. It conferred a benefit, and it stuck around. You know,
Right.
The protease called 3C goes in presumably so it can cleave transcription proteins.
It trashes all kinds of stuff.
Yeah.
It basically makes a big scene. It's like
it makes a scene.
a bull in a china shop.
There we go. We have a title.
Bull in a China shop. Yes. That'd be a good show title. So 3C. It's when you get this big polyprotein, you need a protease because you've got to cut out your individual proteins. But then you can also use that protease as a weapon.
Right.
Yeah.
So people are wondering why the polymerase ends up in the nucleus, because it's actually the polymerase that has the nuclear localization signal. And this paper addresses that. Now, I have a feeling that so they actually say, we're interested in why the polymerase is in the nucleus, and here's what we did. But I have a feeling they did something, and it turned out to explain why the polymerase is in the nucleus. Right? So
You think so?
Yeah, I do.
You sure they didn't just do that pull down
No.
to start with?
So what Rich is referring to is they took an antibody to the polymerase of enterovirus 71, which is a coronavirus sort of related to polio, and they used it to pull down is jargon. They add the antibody to infected cell extracts. It binds to the polymerase, and then they can immunoprecipitate the antibody using beads that recognize the FC portion, basically centrifuge out whatever is bound to the antibody. So the polymerase is going to be bound to the antibody. Then anything bound to the polymerase is going to be there as well. So that's what pull down means. Right, Rich?
Right. This goes back 30 years to Ed Harlow,
Yeah.
right? What did he pull down? SV40 T antigen?
T antigen.
Right.
Yeah.
And a whole bunch of stuff came with it, and he spent the rest of his life studying that stuff. Okay, and it's all, the significant fraction of it is relevant, most notably P53. Okay?
Are we sure about the SV40? Was this first thing?
No, I'm not sure.
I thought that was...
Okay. I was thinking it was some E1A.
That was Arnie Burke Arnie Levine with SV40, T antigen, and P53.
Peter Sarno.
Right? Peter Sarno, yeah.
Ed Harlow got some E1A antibody from me and Arnie Burke.
Ah.
And,
did he pull down P53?
No, he pulled down PRB.
Okay.
It was the antibody to E1A.
All right, so forget I said anything.
All right, you got the general idea.
You're just kind of coming close to something I know something about.
I'm so confused.
No, but
because they don't know that history.
The point is that this whole activity of doing a quote pull down experiment unquote to find out what sticks to your favorite protein is been around for decades,
right?
And is a productive line of investigation.
Just to bring Dickson back into the conversation, it's a form of fishing.
Right. I did a lot of that. I used to call it immunoabsorption, but that was in the old
immunoabsorption.
days. Immunoabsorption.
So they pulled down the polymerase, and they then run what's pulled down on a protein gel, and you get lots of proteins, many. And in the old days, you had no hope of figuring out what they were. But nowadays, you can cut each one of them out and do mass spectrometry.
Yep.
And find the sequence of each and every one of them.
That's amazing.
The magic of multitoff.
In the old days, if you got one protein, you would cut it out and try and sequence it by old methods, and maybe you'd get something. But in this gel, they show a gel here. There are just dozens and dozens of proteins. So I have a feeling Rich and others, they were just doing pull downs with the polymerase to see what attached to it. And what they found was that it attaches to a bunch of nuclear proteins that are involved in mRNA splicing.
Right.
And they developed a story, and then when they tell the story, they say, "Oh, we want to know why the 3D is in the nucleus, and here's why."
Right.
So I just got that feeling. I don't know if I'm right or not, but
well, I'll tell you, there's one of my favorite vaccinia proteins lights up the nucleus like gangbusters late during infection, and I've always kind of said, "Oh man, I don't have time to deal with this."
So the protein or antibodies to the protein.
But yeah, I mean this investigation, going after that is you got to wonder.
Yeah. Yeah. Well, they have a lot of interacting proteins. We don't know how many of them are specific or not because a lot of them just stick, right? But they identified these proteins that are involved in splicing, and we did a TWiV 101 on mRNA splicing a long time ago, right? Or Virology 101.
We did.
And the spliceosome is the structure on which mRNAs are spliced to remove introns, and they're composed of little RNAs and proteins, SNRPs, small nuclear ribonucleoproteins. And Joan Stites is a big SNRP person, right, Rich?
Yes, indeed.
We'll hear about that this summer.
Right. I want to ask her about the story about how she got into that, because there is a fascinating story that goes along with that.
Yeah. Well, we'll do a TWiV with Joan and find out everything, and including stories about Rich Condit.
Yeah.
There aren't any.
They're pretty boring. Yeah.
According to you. All right. So they pulled out lots of these SNRPs, and they focused on one called PRP8. And just before we started, Dickson says, what does that stand for? And I hadn't even thought about that.
It's pre-mRNA processing factor 8.
Is that right?
How descriptive.
Yes.
Good for you, Alan. Wow.
And what else? So mutations in it do what, Alan?
They mess up pre-mRNA processing.
And what else do they do in people? Apparently, they're associated with retinitis pigmentosa.
Yes.
I didn't know that. I thought that was part of the name RP, but it's pre-mRNA splicing. Okay. So this is the largest and most evolutionarily conserved protein of the spliceosome. They describe it as kind of a scaffold on which splicing occurs.
220
don't call it
kilodalton.
Prop 8 because people think that's California politics.
Prop 8? Is that a real bill in California?
Oh, yeah. That was
ages ago when I lived there.
Yeah. Well, not that long.
Well,
that's
a long time since I've lived in California.
Really?
Wasn't that the same-sex marriage ban that finally got overturned?
Oh, you're right. I'm thinking of something completely different. Yes.
Mm-hmm.
Yep.
California, I mean, besides even outside the moving industry, California is full of props. They're always doing ballot initiatives of various sorts.
So this thing is 220 kilodaltons, which in the world of proteins is like really big.
Yes.
We're talking top of the gel, dude.
Oh, that's like EIF4G, P220.
Yeah, it's 220 kilodaltons.
Wow. Yeah, that's a big protein.
Yeah, it's big.
Holy
And
cow.
it does a lot.
Big platform.
I'll say.
So
this review, who dug up this review?
I
on
just,
PRP8.
I just found it, yeah.
It's got a couple of figures in it that show all of the things that this interacts with and all of the yeast mutations that affect its activity. It's amazing.
This is like the splicing protein that's,
I'll say.
everything goes through there.
I guess that's why they picked it, right? Because they had a lot of
I
proteins.
suppose if you get, because they got a lot of hits to interesting stuff. But of the interesting stuff, my guess is if you're into splicing at all, this is the most interesting because it's, right there in the thick of it.
So now you have a putative interaction between the viral RNA polymerase and this PRP8, Proposition 8.
Right.
And now, since you have antibodies against both, you can do a whole series of things which people do all the time when they have interacting proteins, is you can figure out what part of each one interacts with the other. And you can do this in a very cool way by, you can use antibodies to immunoprecipitate one protein and then run that on a gel and do a Western blot with antibodies to a different protein, right? So you can pull down with polymerase and blot with PRP8 or vice versa. You can, and you end up getting lots and lots of gels. People who love running gels go crazy with this stuff. So basically, they confirm the interaction, and they make little truncations of each protein, and they can figure out what part of each are interacting. So I think it's the C-terminus of PRP8, right, is interacting with the fingers domain of the polymerase. Polymerases, Dickson, have been described as being analogous to a right hand with fingers, thumb, and palm domains.
Look at that.
And in the palm is the active site. Of course, it would be there, right?
Naturally.
You wouldn't be giving the finger to an active site. No. I said that for Alan, because I knew he was going to say that.
Yes.
Right, Alan?
Yes,
you
I
were
was.
You were thinking.
I give that a thumbs up.
Yeah, that's what you were thinking.
I give that a palm down.
Okay, so that's how they interact. And you know what? This is quite interesting because one experiment they never do.
Go on.
Whenever you do this, when you establish what parts of two proteins interact, what you would like to do is abolish the interaction and ask what happens.
What happens, right?
Yeah.
So you could abolish the part of the fingers that interact with PRP8, put that in a virus, and then infect and see what happens. They never do that, and I'm really sad because...
You never cut off the hand that feeds you.
That's another show title. Oh, well, they didn't do it. We have to save it with that. Anyway, so they describe...
Well, I was wondering whether there would be... Well, I suppose you can make an infectious clone, can't you?
Yeah, absolutely.
So you can even put a lethal mutation into the virus, even if it's lethal. You can do that and do the transfection and ask what happens.
Yeah. Hopefully you could make a 1 or 2 amino acid change in the fingers and it wouldn't kill the virus, and then you could ask, is there any consequence of shutting off or interacting with PRP8? We don't know what the function.
They also have a number of in vitro assays that they could have done here with mutant protein.
I mean, there's a lot of work in this paper, so
it's huge.
That's a separate paper, but I'm burning to know the answer, and I'm sure we'll find out one day. All right, so next they look to see if where these proteins are in an infected cell. So they infect cells with the virus, and then they can stain the infected cells with antibodies. Different colored antibodies, green for one and red for the other, and you can see that the polymerase enters the nucleus, as it should, and it co-localizes with PRP8. Dickson, how would you tell if it's co-localizing?
Well, the two colors would overlap and change color then, wouldn't it?
What would it be if you had green and red?
Green and red. Let me think.
Hard. Think hard.
Bluish.
I really thought you were going to get it. No, yellow, Dickson.
Yellowish?
Were you
I said
kidding?
yellowish.
Were you kidding? I figured you would have done this in your career, Dickson.
I've done it a lot. I took watercolor classes for 8 years.
Yeah, that'll do it. Okay, so they co-localize.
I want to just a little.
Of course.
I couldn't help but notice they do a time course of this co-localization, and I was entertained by the development of a cytopathic effect. The cells go from flat to round. That's cool.
Yes, that's typical.
I mean, I get off on this kind of stuff, right?
Yeah.
And also, and they comment on this, and this is interesting, is that late during infection, the PRP8 actually exits the nucleus and winds up in the cytoplasm.
Interesting.
Right.
Yeah.
I wondered about that.
So the virus, polio at least, messes the nuclear pores up. So towards the end of infection, everything comes out of the nucleus. So that may be what's happening there.
Okay.
I don't know if EV71 does. I don't know if anyone's looked at that, but that's what happens with polio. They take out the nuclear localization sequence from the polymerase, and sure enough, it does not go in the nucleus any longer. So that confirms that that's doing what we think it does.
That's another mutant that could be used to study this whole thing, because they have some in vivo experiments later on to show the effects of the polymerase, and they ought to do that without the nuclear localization signal. So,
Gosh, someone must have done this with polio. It's been known for many years that there's an NLS. I'm sure it was done. I wonder if that virus would be infectious or just very sick. I don't know the answer. Do you know the answer, Dickson? No.
I am, but I'm not saying.
Okay. Alan Dove, can you look it up while we're chatting here?
I'll see what I can do.
Question of point.
Because you're an old coronavirologist,
I have a question from the Peanut Gallery.
Yes, sir.
Is it the same nuclear localization signal that all proteins use, or are there different nuclear pores that allow different proteins in?
Well, they're kind of conserved, highly basic. Like this one is KKKD, and there's short and longer NLSs. So they're I don't know that they're different pores, right? I think you can take an NLS and put it in a different protein, and we'll get in the nucleus.
Okay,
right.
So they all use the same basic.
Not exactly the same, but
I meant basic.
They're different types of NLSs, right? They're simple and complex. Okay,
but they're all going through the same pore,
and that's the extent of my knowledge.
Okay, just out of curiosity, that's all.
It's okay. You can ask questions. All right. What's this interaction doing? That's the next question. So since PRP8 is involved in splicing, they look at splicing, right?
Yeah.
And they have an in vitro splicing assay where you make a pre-mRNA, you label it with P32, and you mix it with a nuclear extract, and you incubate it, and it will normally splice. You have the pre-mRNA, which is longer, and it goes to a shorter mRNA, and you can also see the Lariat, that is the intron joined to itself, a circle, right? And you can see that on the gels that they run. If you add polymerase, it inhibits this.
So the Lariat is an intermediate in the splicing reaction. The first part of the splicing reaction is to cut on one side of the intron, and that is a concerted reaction that involves making this branch structure where the 5' residue on the left-hand side of the intron forms a bond with a base in the middle of the intron to form a branch structure that, if you think about that, it comes out as a kind of a loop. They call it a lariat, and that's an intermediate form. And then ultimately, the 5' exon then joins to the 3' intron, and the intron is excised still in a lariat form. And you can resolve all of the intermediates and precursors and products on a polyacrylamide gel in the course of the splicing reaction. So you can see whether you've got intermediates and stuff. And they show that this protein inhibits the splicing reaction, and the lariat form intermediate accumulates.
And just an update on some quick PubMed searches. A search on poliovirus 3D NLS only gives me 3 papers, one of which is this one of which is a review, and the third is by Sharma, Raychaudhuri, and Dasgupta from 2004, published in Virology. The the full text is on Elsevier, so it's paywalled. But just looking at the abstract, it looks like they did mutagenize the NLS. They say that prevents nuclear entry of 3D and 3CD in the infected cells, and that 3CD can be detected in the nuclear fraction as early as 2 hours post-infection.
But so the virus is infectious, right?
I don't know if they looked at the effect of this on the virus, really. I mean, they seem to be most interested in proving that this NLS is functional. All
right.
And
so I don't know. I think it's not been looked at very closely.
Not been looked at, yeah.
I tried to find if there was anything about EV71 and nuclear pore, and I only found that the 2A protease seems to cleave NUP62 to inhibit nuclear transport, but I don't know if that would have a downstream effect on pores.
And broadening the search to picornavirus 3D NLS brings up 6 papers, the 3 I just mentioned, and then a few others which basically are, let's see, somebody looking at encephalomyocarditis virus and showing that 2A and 3B/CD localized to the nucleus to inhibit transcription, mRNA but not RNA transcription.
Okay.
Surprising. Can you imagine what TWiV would sound like if it was recorded 20 years ago? Wait a minute, I have to run out to the library.
Exactly.
Where's the library? It's over
Hold
there.
the show. I've got to take the elevator.
Exactly.
Hey, Alan, send me somehow, maybe in the chat or something, the PubMed reference to that Sharma paper.
I can do it. I'm right on it.
Oh, okay.
I can send you the paper.
It's that real-time collaboration.
Instant gratification.
It's wonderful. All right.
Splice it over to him.
So 3D inhibits splicing in vitro, and they looked at other picornas that have NLSs, polio, Coxsackie virus B3, rhinovirus type 16. Poliovirus 3D inhibits splicing in vitro, but neither Coxsackie nor rhinovirus 3D inhibited splicing. It's interesting because they have NLSs, and presumably these proteins go to the nucleus, but they don't inhibit splicing. So maybe going to the nucleus is not the key, right? Maybe splicing isn't the ultimate target. Who knows? Or maybe there are different targets. Am I making sense?
Yeah.
I think so.
When I hear silence, crickets, I wonder if they say, what is he talking about?
Someone had to nudge me to wake me up.
All right.
That was a joke.
The 3D polymerase of EV71 in polio, remember we showed in this paper, EV71 polymerase associates with PRP8 C-terminus, but the polymerase of Coxsackie and rhino do not. So that explains why they don't inhibit splicing, right? So that interaction is important for inhibition of splicing, apparently. All right, so that's all in vitro. Now they look in infected cells. Mm-hmm. You infect with virus and you ask, is splicing inhibited? They first use a reporter, which is a plasmid containing a promoter, and it's going to generate an RNA in the cell. It has an intron in it, and you can track that specifically. And they show that, in fact, enterovirus 71 infected inhibits splicing of that reporter at 4 hours post-infection, and you get pre-mRNA accumulation and reduction of the mature mRNA. And if you overproduce PRP8 in those cells, you can restore splicing. You like that one, Dickson?
I love it.
Yeah. And then they also looked at an endogenous gene. They looked at the mRNA encoding nucleolin, and that, in fact, its splicing is inhibited also by the polymerase. So it happens in infected cells. So the interaction with PRP8 apparently inhibits splicing. Everyone convinced of that? I think.
I'm convinced of that, though I would like to see those experiments with the mutants.
Yeah, that would be good to see if this is biologically relevant for the virus, right? Yes,
right.
I mean, that's the question, because it's an interesting effect, but if it doesn't help the virus...
So,
right?
I mean...
the problem is
you could easily have a situation where you could make the mutant, and I mean, the mutant ought to show the defects in the splicing. The mutant itself wouldn't necessarily be defective because this virus is doing so much to mess over the host, okay, that this could just be, another thing that it's doing. And you wouldn't necessarily see a phenotype unless you basically stuck it out in nature and gave it a couple of million years, okay, see if it
Not
did
even.
better.
If you just stuck it in vivo, you might see differences.
But maybe they did that and didn't see differences.
Maybe. There is a mouse.
We don't want to go all reviewer number 3 on them. It is good paper, but
reviewer number 3. You get that, Dickson?
Yes, I got that.
The thing is, if you knock out the nuclear localization signal, right, that's too broad because it may be going in the nucleus for some other reason besides inhibiting
it's carrying
splicing,
the 3C portion.
3C,
exactly. There's too many things at once. So I like to knock out the PRP8 interacting part.
Yes.
See what happens there.
Yep.
So the next thing they did is ask what mRNAs, splicing of which mRNAs is inhibited, and can we learn anything from that? So they immunoprecipitate PRP8 and then look at the RNAs that are associated with that. The idea being, I guess, that the splicing is inhibited, but those RNAs, the pre-mRNAs, are still stuck on the spliceosome. And so they bring all those down, they sequence them all, which you can do these days very nicely. Another great technology, you can do massive sequencing and ask what kinds of genes are associated with that. And you can do those all in the software now. You get lots of transcripts, and you can ask what functional groups are these are the encoded proteins in, and they find that if you compare the virus-infected versus mock-infected cells, the mRNAs whose splicing is inhibited are involved in cell growth, proliferation, and differentiation, which to me seems like almost everything.
It's pretty
the
much.
cell's genome is down there, yes.
This seems like a pretty blunt instrument by the virus.
Right, there you go.
Yeah.
So,
which makes sense because this PRP8, it's such a central regulator and mediator of all this splicing activity. If you screw that up, you're going to shut down all kinds of stuff, and that seems to be what the virus is aiming at here.
I mean, you don't want to kill the cell too quickly, though, right?
Well, no, but it's just shutting down splicing.
So there's still some spliced mRNAs.
There's some spliced mRNA. There's still some translation going on. There's still and polio. And picornaviruses are generally pretty quick.
Yeah.
Anyway, I mean, it doesn't need weeks to kill the cell and make its progeny. So it's getting the work done in the cytoplasm, and it just needs to make sure it's not going to get a lot of interference.
Yeah. I mean, you still need energy. You need membranes. So you don't want to trash everything in this cell.
Not quickly, anyway.
Not quickly. I mean, this is at like 4 hours post-infection, which is almost at the end of the cycle. So I don't really know what's the purpose of this, because the virus, at least polio, shuts off translation. It shuts off RNA synthesis in the cell. It shuts off vesicle transport. So, I mean, they say that, maybe those things don't cover everything. Just make sure that everything else is done. But it seems to me that it's kind of late, and I'm just not sure. It's really an interesting finding.
So when do you get mature?
So 4 hours is late for EV71?
So that's, for polio, it certainly is late.
Mm-hmm.
And
when do you get mature virus?
Well, I don't know for EV71. For polio, after 2 hours, you start to get viruses.
That's
produced.
remarkable.
And they're using a high MOI, which is 40 PFU per cell.
Wow.
And for polio, that is 2 hours. You start to get virus out. So I don't know what the cycle is for EV71, but it's a pretty impressive enterovirus. So I would guess it's similar.
Yeah.
There may be antiviral factors that it's trying to stop.
Could be
the production of.
Could be. That's why we'd like to knock this out and see, right?
Right.
They do talk in the very end of the discussion about some RNAs that are not, or that are increased upon EV71 infection for chemokines, protein degradation, complement, pro-apoptosis.
Right.
And so that suggests they're escaping the transcriptional regulation, and I guess it means they're escaping the splicing thing too. So.
I found it interesting because I don't know of another example of this kind of specific interaction and inhibition with the splicing machinery. Right? Do you know any, Rich or Kathy? Do other viruses do this?
I can't. There must, but I can't think of examples right offhand. There's another question for Joan. I mean, the first thing that comes to mind is the Ebers, right? But that's a little different.
Yeah.
Well, there's viruses that do things to regulate alternative splicing.
Right.
Adenovirus does that, for example. So they do make things that interact with the splicing machinery.
Right. They say this is the first study demonstrating that a cytoplasmic RNA virus can use its polymerase to alter splicing, which is a very specific statement.
Yes.
Extremely
Right. So
specific.
it implies that maybe DNA viruses are already known to do that.
Well, now most papers come to fairly specific,
Yes.
unique results.
Anyway, if you had found this many years ago, it might have been a way to discover the spliceosome machinery, right?
Sure.
That's discovered a long time ago, but this is just another way that viruses could help you do that. But I found it interesting, this specific interaction, and I mean, there are a lot of questions unanswered, and hopefully they'll answer them.
Would you learn anything if you could use this virus to infect cells that were suffering from the retina pigmentosa defect?
That's interesting. I don't know exactly what that defect does to this protein, but let's say it really alters the protein, right, Dickson, so that the polymerase will no longer interact.
Exactly.
That could be interesting, phenotypically interesting. Yeah.
So that gene probably is known, the mutant. That would be Rich's mutation study that would work out pretty well.
Maybe. Depends on the mutation in that those cells. Yeah. It's very much like how you can use cells from people with Niemann-Pick disease, and they don't allow Ebola virus entry because the receptor is not there.
Right.
Well, nowadays you could also just take ordinary cells and use CRISPR-Cas.
Yep, absolutely.
Muck around with this thing at will.
So the thing is, if you CRISPR'd out the PRPA gene,
not the whole thing, right?
I'm not sure the cells would be happy.
You'd want to be more surgical than that. Mess up maybe just the binding site for 3D, which may mess up other things, but
Yep.
yeah.
All right, so that's it. Any other insights?
I was still a little puzzled by the fact that, they see the 3D and the PRP8 co-localized at 4 hours after infection in the nucleus, and then later they see them in the cytoplasm at 8 hours after infection. And they say, the interaction in the nucleus disrupts the splicing, whereas the interaction in the cytoplasm may support the function of the 3D polymerase during viral infection.
Yeah.
And I wonder do you really think so? I was just thinking it was just either, like you said, the leaking out, or, that's where it's being translated, and so that's where it associates, or it can associate there. I don't know.
It's kind of late.
Yeah.
I was thinking 8 hours after a poliovirus infection, everything is basically done, right?
It's pretty much. I mean, if you really need PRP8 to get RNA polymerase active, that's just too late because it's active way before that.
Yeah.
So, I mean, you can find out, right? Just disrupt the interaction and see what happens. But I think you're right, Kathy. It's just leaking out, and that's all there is to that.
See, I just pulled up a kinetics of enterovirus 71 replication, and they show EV71 RNA seems to peak at 6 hours.
Maybe a little later than
Okay,
polio.
so a little later, polio.
Yeah. Polio spoils me.
Yeah.
It's like a phage.
Virus packaging peaks at 12 hours.
It sounds like there's no excuse for not doing a lot of work with polio because you could do 3 or 4 experiments a day.
Hey, Rich, when you worked on phage, was the generation time like 20 minutes or something?
20 minutes. Matter
Holy
of fact,
cow.
we used to do our infections at 30 degrees instead of 37 just to slow it down. And I had so much fun with these experiments when I did pulse labeling experiments because I would set up water baths with infections going and take time points every 2 minutes from 4 different cultures, and they would be pulsed and then quenched and stuff. And it took such intense concentration for a period of about 30 minutes, and then it was all over. Wow. And you knew the answer the next day, and you thought up another experiment and did it the following day,
Oh, goodness.
My viral entry studies when I was doing my thesis work were kind of like that. I did time points every 10 minutes, and for a couple of hours it was just you're doing that, and then throw a bazillion vials in the scintillation counter and go have lunch.
Right.
Yeah.
Just as a way of showing perspective differences, I'm a parasitologist that worked on trichinella. It was a month-long life cycle. That explains a lot, I bet.
Yeah, it does.
Plaque assays with T7, you'd either stick them in the 37-degree incubator for 4 or 5 hours, or leave them out on your bench overnight. If you put them in the incubator overnight, the plaques were too big the next day. You could take them.
That's great.
Wow.
That was wonderful.
Wow.
Listen, I have a little update on our previous conversation.
Mm-hmm.
There are early 1980s papers with Harlow and Lionel Crawford describing the interaction between SV40T antigen and P53.
Mm-hmm.
But then in the mid to late 80s, he switches to E1A. And there's a nice paper here, Adenovirus Early Region 1A association with cellular polypeptides. And then he moves on to papillomaviruses. So he made a small industry out of this pull-down sort of thing.
Yep.
Right. And I'm trying to figure out when the Sarno-Levine P53 paper was. Maybe it's a little bit later, looks like. 82. Cell paper in 82. Yep.
What was I going to say? Oh, it's gone.
I have something else to say. I was thinking of Prop 13, and the fact that they went back to early numbers confused me.
Right.
So Prop 13 was 1978.
Okay, it's California then.
All right, one more note from the American Society for Microbiology. This is from their education department. They would like you to know that the Journal of Microbiology and Biology Education, which they affectionately call JIMBY, has published a special section devoted to scientific ethics. And this is guest edited by Dr. Beth Fisher from the University of Pittsburgh, Dr. Frederick Grinnell at the University of Texas, and Dr. Michael Zygmunt, University of Pittsburgh. It's a 45-article section with a comprehensive look at ethics in science, including responsible conduct of research, RCR, ethics in science education, scientific publishing and authorship, and ways to incorporate ethics into STEM education. And it also is essential for the ongoing education of active researchers and mentors. Gives articles about why you need training in RCR, ways to incorporate and evaluate RCR training, and the potential influences, promise of funding, research competition, and authorship that may compromise research integrity. These are all things we've talked about here. So it's a pretty cool issue. And you can find it at jmbe.asm.org. Special issue. Check it out.
Cool.
All right, let's do some email.
And you can crack open a bottle of Jim B. and... Oh, no. Never mind.
No, that would be unethical.
Ooh. You listen, Dickson.
He does sometimes.
Stop it, all of you.
We're not making you feel bad, are you? Are we?
That's hard to do, actually.
Kathy, you want to start with Sharon?
Sure. Sharon writes, "Hello, TWiVers. Thank you for discussing career choices on TWiV in addition to the science. In episode 323, in the midst of a heated debate about whether or not to do a postdoc, Vincent mentioned that he likes the idea of staff scientists, and I agree. However, from the science gossip that I hear, most PIs are not hiring staff scientists. I hear this is because they cost more. Would you all comment on this a little further, please? Do you think most PIs share Vincent's thoughts regarding staff scientists? Thanks in advance for your answers. Best, Sharon." And she's a postdoctoral fellow at UT Southwestern.
I think we can't speak for everyone, but we could certainly have people write us and let us know what they're thinking. I think it's a great idea. I know that John Udell at NIH was one of the first people who suggested this as a way of changing. But I don't know. Do you guys have any insight?
Well, if you were going to do it off of an NIH grant, our students cost us, if they're not advanced to candidacy and we're paying tuition and their stipend, they cost us around $60,000. But once they advance to candidacy and then their tuition goes down and so forth, so they're a little bit cheaper than $60,000. But we're talking staff scientists here that have PhDs, so I'm thinking that this is going to be hard on NIH budgets as they stand now.
And these are going to be permanent positions, which, ethically, you really ought to be giving these people benefits as well.
Yeah.
So the
they would.
total compensation package is going to be definitely into the six figures for these people if you're paying them fairly.
Yeah.
Yeah, here the staff scientists start at 60, and then you pay fringe on top of that, which is 30% for their benefits and so forth. And there's a range, I think it's 60 to 80 or something like that. So, I don't know. I think it's worth it because you have a long-term association of professional people who are highly trained and can get results. I think it's good. For them, it's good. For you, I don't know.
Basically, for the last 10 years, and even on 10 years for sure, I've run my lab with two staff scientists and occasional and postdocs, and me and a staff scientist and a postdoc and supplies sucks up a whole 10-module grant.
Right.
Okay, so that's that. But I don't regret it for a minute. It's a nice way to run a lab. It's just that a 10-module grant, it's expensive. And even though, I mean, by the standard we've just discussed, they were paid fairly, it's still, these people have got PhDs and a lot of experience. It's not, I think we ought to pay them better than that.
Yeah, I think to get this to happen at any reasonable scale, there needs to be a rearrangement of the whole grant structure. Because right now, what you're saying is it's nice to have staff scientists, but to advocate that as a solution to the whole postdoc problem, you'd need a lot more of these people, and PIs would frankly have to act against their own self-interest. Because you certainly get two postdocs for the price of a staff scientist, by any reasonable structure. And if the postdocs are going to produce even close to as much as a staff scientist, you are going to produce more papers by hiring more postdocs.
But that's the point, right? We have to act against our self-interest.
But I think that's not going to happen. It's lovely to have that as a notion, and some people who are particularly progressive about it and who are privileged enough to be progressive about it will go ahead and do that. Tenured faculty who don't have a lot to lose, they don't have to worry about whether they're going to get tenure, might have the luxury of going to one staff scientist and a postdoc, and you produce the papers that come out of that. But if you're gunning for tenure, I don't know that that's going to work, and I think it would be a risk to take that I think is a lot to ask.
So we have to change this scientist, we have to change publishing, and we have to change tenure all at once.
Well, we don't have to change tenure, but we have to change the way. I think the NIH could fix all of this with a few strokes of the pen, because the reason you want graduate students and postdocs in your lab, I mean, it's obviously you want to train them, but the productivity that they're generating is based on the NIH grant allocation formulae, right? And if those allocation formulae change and they say, okay, we're only going to pay for one graduate student per PI per grant, well, guess what? You suddenly have a much lower need for graduate students, and your program will automatically contract because there won't be places to put these graduate students. So that shuts off the oversupply of PhDs, and then you do the same thing for postdocs. You say, look, we're going to pay for one postdoc or two postdocs per PI. But you know what? We'll give you an unlimited number of staff scientist positions within the parameters of your grant. Now, you're still competing for an amount of money that'll pay all those staff scientists, but the big labs could staff themselves with staff scientists. Those positions would then exist, and you could sop up some of this massive surplus of labor that's floating around.
Well,
go ahead.
I just want to ask people, let us know what you think.
That's my off-the-cuff proposal.
The PIs out there, let us know.
Kathy, did I hear you say that a starting graduate student costs you $60,000?
It's about that. For instance, if they're a second-year student and they haven't advanced to candidacy and they're not on a training grant, then by the time you pay their stipend and the tuition
Yeah.
here.
It's about the same here, yeah.
Whoa.
Yep.
And it includes health insurance.
Right.
Yeah.
And
Right,
so
but
when they
the
advance
tuition, go ahead.
when they advance to candidacy, how does that affect it?
Well, their tuition goes down, but it's more like what is it, $5,800 a semester? Then, yeah, so it's less.
So
Something like that.
So the bottom line at that point is how much? 40, 50?
Say 30 plus 12, yeah, 42.
So 42?
Well, 42 to 45 with the health insurance.
Wow.
Yeah. Yeah, it was a big change from someplace where I was before that there was complete tuition remission, so we didn't have to pay tuition for our students.
Yeah,
So-
but the tuition is basically a scam the university runs to get more money out of the grants because they know that the students are not going to be paying it.
Right.
Yeah.
Amazing.
All right. Let's continue. Rich, can you take Todd here?
Todd writes, Dear TWiVocateurs, on TWiV 323, you read a letter from Len, who recounted his experience on Twitter of rapidly being blocked by an anti-vaccine ideologue. He asked you for advice, and the consensus was along the lines of, don't bother. Masochistic advocate for public health that I am, I actually engage some of these folks from time to time on Twitter. However, I enter into it understanding that more likely than not, I will not change their mind. Instead, my goal, both on Twitter and elsewhere on the web, is to be an example for those who may be watching that are confused or on the fence. I strive to present a calm demeanor and stick to the known facts as far as they're currently known. The result is typically that the anti-vaxxer gets more and more unreasonable. If they do not block me right away, I eventually get around to the question, what would convince you that your position is incorrect? This generates one of two responses. They block me, a trend that has increased with the recent negative attention the anti-vaccine movement has been getting, or they respond that nothing will change their mind.
Great.
My hope is that these interactions will show those who have questions on this on the one side are reasonable people willing to engage and talk about the questions, even the hard ones. And on the other side are entrenched ideologues who would rather vilify pro-science folks and shut down the conversation. Just my 2 cents on the subject. Currently 18C minus 8F, and we're rivaling Buffalo, New York for the total snowfall this year at nearly 8 feet of snow, probably more than that by the time you read this. Best, Todd. P.S. The neighborhood Yeti is wondering when you'll do an episode on cryptozoonotic illnesses. Is Todd writing from Boston?
Maybe. Well, I guess you'll have to Sasquatch for that episode.
Dickson, that's for TWiP cryptozoonotic, no?
Well, no, it's cryptozoan because it's a cryptic. It's a, yeah, could be. So David writes, Dear
Wait
TWiV
a minute.
hosts-
Wait, before you go.
I'm sorry.
Cryptozoonotic means what?
It means hidden life.
So it could be viral?
The Yeti is part of that. You're imagining things that have yet to be proven. So cryptozoonotic is a
Nessie.
reference to a mythology associated with creatures like the Kraken and
Nessie. And,
the Yeti. I keep going. Yeah, that's exactly right.
the Kraken actually exists, but the
Not to cryptozoa.
no, not in the way that was imagined.
All right. Thank you, Dickson.
That's okay. Carefully. I'll read the next one now.
All right.
David writes, Dear TWiV hosts, an article appeared in the New York Times that received the following comment from a reader. Quote, your characterization of the anti-vaccination propaganda as an unreason at its culmination is not only unjust and absurd, but even ridiculous and tends to show how much ignorance of the facts. That's what they wrote. That's what I read. You never could and never will be able to prove beyond a doubt your statement that vaccination long since proved its utility and its harmlessness for this is not so. If you had followed up and studied more the disastrous results caused by vaccination, you would perhaps have less to say in its favor. The comment and article appeared in the New York Times in 1910. Unfortunately, I wasn't able to find the article to which the comment refers. I recently accidentally stumbled upon some newspaper articles from the late 1800s and early 1900s on the anti-vaccine movement. With slight changes in the language, the stories are not really different from those of today. I guess things haven't really changed. On TWiV 323, there was some discussion and links on the problems with using data to convince people of the benefits of vaccination. I like data, but not everyone finds it helpful or convincing, unfortunately. The anti-vaccine movement relies on personal stories. We can do that too. I always reveal that my kids are fully vaccinated, and I like to send people to a website called shotbyshot.org. People's personal stories of their child dying of a vaccine-preventable disease makes a pretty powerful and emotional argument in favor of vaccination. I wanted to recommend this video in the New York Times, and it is quite good and features Seth Nunkin, whom you had on TWiV some time back, and there's a very long reference to that. I don't know the temperature outside, but it's 11°C in the hallway and 16°C in my laboratory, so I hope they fix the heat zone. I suppose I need a warm water bath for my room temperature incubations. Yeah. Cheers, David.
Yeah, he sent an image of that comment, which is pretty funny. You can actually find from Jenner's time anti-vax
absolutely
pictures, right? As soon as we started, the anti-vax people jumped in.
The beginning of vaccination was fraught with all kinds of controversies and the need to prove by example. The one I remember best was the one on smallpox in the United States, but there are lots of others.
Sure. This shot-by-shot site, by the way, looks quite good.
Yes, I checked it out last week. It is really good.
It's like a multi-disease version of the Families Fighting Flu approach, and I think that can be extremely powerful in those types of discussions.
Yeah, that ought to be prominent on the TWiV page. Maybe give him a pick of the week for that.
Sure.
Yeah.
Yeah, it's a pretty easy URL to remember too. Shotbyshot.org.
That is good. Nathan writes, "Hi there. Just sending a suggestion for your website that would improve its accessibility. Below each TWiV episode, you should show the links. Basically topics for the episode. My suggestion would be to have the hours, minute times of each topic discussed in the podcast, which a website viewer could click, bringing them to that part of the conversation. If a viewer has a particular topic of interest in the episode, it is hard to find the specific point in the podcast. Currently, one can only resort to trial and error, clicking on general parts of the small podcast bar in order to find a specific area." Yeah, it's a great idea. I just don't have time to do it.
Right. We need you need a staff person.
I need a person.
You need
you need people, Vincent.
Yeah,
a staff scientist.
If you can remind me of this when we start taping the next episode, I might be able to do something with that.
Well, the problem is that
the tape time in the episode is
the tape
the
time
same
and the episode
as
time are not the same. I know,
so
but
the final thing is
okay, right.
I mean, I guess I could scrub through quickly when I finish it. So I could try it, but
or you can
It's just I have with my text editor set up. I have an elapsed time timer. Little script I put together because when I'm interviewing people, I like to mark the time. I don't do literal transcripts, but I put in timestamps in my notes so that I can go back and pick out quotes from a tape.
That's good. Yeah.
So I kind of already do this as part of my workflow for other things, but it would have to be something that would be part of your editing, where you note the time when certain topics come up, and I don't know that that's going to fit in.
If you could sync the first one to a certain time in the actual podcast, and then Vincent would have to do the arithmetic to adjust all the other ones.
Right.
But
So I guess we'd want like follow up, the paper, email, and pics, right?
Right. At a minimum, yeah.
You don't really need much more than that, right? I guess that we usually not lately. We've just had one paper now, so that makes it easier. All right, let's do one more, Alan. We're up to you.
Okay, and we're doing Charles, right?
Right.
Yes.
Charles writes, "Dear TWiVers, hello from sunny Adelaide, Australia, where it is currently a sunny 31.1°C with 32% humidity and a pleasant 8-knot northerly breeze. It was with some interest and probably pretty dry, right? Weren't they having a huge drought?"
Oh, yes.
Yeah. All right. It was with some interest that I listened to TWiV 323, a skid loader full of viromes. As someone living with Crohn's disease, the volume of research that is currently being published on the gut microbiome slash virome and its effects on diseases is as diverse as MS and ulcerative colitis warms what's left of my bowels. It appears to me that research into the human virome microbiome has really been ramping up in recent years, but I'm not sure if that's actually the case or whether science writers have only started covering this area of research in recent years. Thank you for your podcasts and keep up the good work. You do a great job in showing that science is fun and in communicating current research to a general audience. My inclination and training is in the physical sciences, so the details often goes straight over my head, but that was often how I felt at university, and I somehow managed to graduate again. P.S. Have you done a TWiM podcast on the human gut? I scanned through the titles and didn't see anything, but with some of the titles, it's a bit hard to tell.
Hi. Yeah, I think we did.
So research on the microbiome has definitely ramped up a lot in recent years, and it is driven by the latest burst in this is driven entirely by the advancement of high-throughput sequencing technology.
That's right.
So you can now, we've known for decades, if not centuries, that there were lots and lots of microbes out there we just couldn't culture. Right. But now there's finally a set of tools that allows us to find them and study them, and that's why all this research is coming out now, because now we can actually see them.
Yeah, we've done several TWiMs on the gut. We'll see if I can put some of the links in the show notes, Charles.
You'll have to go through and do a gut check.
I will. Let's do some picks of the week. Scrolling down through these emails here.
I didn't send you one, but I do have one.
I have a suggestion about, just because it came up about the vaccine thing, to do a measles sit rep. Yeah. And so there's, clicking on it's not working for me at the moment. There we go. The number of cases from January to March 2015 in the U.S. is 173 in 4 outbreaks.
And the most in California.
Right.
Disneyland.
Gee.
All preventable.
Yep.
There are some states with none.
Right.
Hey, measles, herd immunity is fading. What are you going to do?
Right.
That's pretty good. Yeah, nice. Kathy, what do you have as a pick this week?
Oh, so I picked something that Isabella sent me. It's Ancient Trees, and it's a selection of black and white photos of really old trees that are part of a book by this photographer, Beth Moon, and they're just really striking, interesting photos.
Wow. Now, Dickson, this is the book you should have published.
I
bloody
I would have loved
flowers.
to. Would have loved to have just gone to those
These
places.
pictures are just absolutely stunning.
Wow.
I love really good black and white. Yeah. Oh, this one looks like an infrared.
And the trees are just wild.
They're dendritic.
Yes.
They are.
Yeah,
baobabs.
cool.
Those
Nice.
are baobabs, I guess.
I love the ones, too, with, there's this one with the trees flanking the sort of medieval-looking door.
Yes.
And another one,
The
the
Hobbit.
trees sort of... Yeah, exactly. It looks exactly like the Hobbit. And there's another one with a tree kind of eating some ancient structure.
Yeah.
It's just amazing.
My wife and I once had the privilege of visiting Siem Reap, and then we went over to Angkor Wat and to Angkor Thom, and that's where they filmed Tomb Raiders. And the trees are literally reclaiming all of those structures. They're just eating them alive, and it's quite impressive to see that up close.
Dickson, did you say you had a pick?
I have a pick, yes, I do. And it's, regrettably, Carolyn Porco is retiring from NASA. Does anybody else know what I mean by that?
Nope.
No.
Carolyn Porco is the head scientist in charge of the Cassini probe. So she's the one that sends back all those beautiful pictures of Enceladus and the rings of Saturn and the storms that occur and the lightning bolts at the top of the of Saturn's poles, and the northern pole of Saturn is a hexagon. It's not a circle. You know, you see these swirling clouds. It forms an absolutely perfect hexagon. It's stunning photography, and of course takes you right to the site. And sometime soon, I think this summer, the Cassini spacecraft will pass within 30 miles of Enceladus. So I had the privilege of meeting her once at a meeting, and I told her how much I admired her work, because she really got up there and told you how exciting it is to be the managing scientist for all these pictures when they first come in. She's the first one to see them and to try to interpret them and everything else. And she's moving on to greener pastures, I presume, at Berkeley, and as a senior astronomer in their department. So she sends out a letter. You can subscribe to the Cassini spacecraft news, and indeed she sends you these little bon mots. She's quite a charming and engaging scientist. Kathy, she's one of the leaders in women in science movement that I truly, deeply admire. So I lament her loss to the Cassini program, but I guess people at Berkeley will really benefit from her going there.
Nice. Porco Rosso.
See, Porco, I would have picked that name anyway because of my association with trichinosis.
Porco Rosso is a movie. It's a cartoon.
Yes?
By Hayao Miyazaki.
Huh?
Probably means nothing to you, Dickson.
You're correct on that one.
And boy, are you missing something.
Have you seen any of Miyazaki's films, Dickson?
No, it doesn't sound
Name
like
another one.
Spirited Away, My Neighbor Totoro.
The answer is no.
Dixon is not an animation guy.
I'm totally... no, that isn't quite true. I do like Ratatouille, which was a great movie.
See
and I
Spirited Away.
The Triplets of Belleville was one of my all-time favorites.
Oh, yes.
Oh, yes.
Love
definitely.
that.
So that's come on.
Do see Spirited Away. That's
Okay,
the one
I will.
starting.
I have lots of good stuff to look forward to.
Alan, what do you have for us?
My pick is a new collection that PLOS has put out. Now, it's not a new journal. It's a collection, a feed, essentially, of papers that have been published in PLOS Journal, or PLOS One, the big one. And I would have named this PLOS None, but
yeah, nice.
It's a collection of negative results. And this is a problem we've talked about a bit on TWiV in the past. You're doing experiments and you're trying to test your hypothesis, and you get some results that the experiment just it doesn't work out. It doesn't really tell a nice story. It's a negative result, and so you file it away because nobody's going to publish it. Well, PLOS One, having lowered the bar for science publishing so that technical competence is basically all you need to meet there. They publish a whole lot of stuff, and thousands upon thousands of papers daily, it seems, is just this fire hose of papers, including a lot of papers that are negative or inconclusive results. So
I've got a data book at home that I can.
They are now. That's one step in getting these things out there, and the complaint about the traditional handling of negative results is this is stuff that didn't work in your lab, and probably somebody else is going to end up repeating the same thing and wasting money and time on the same effort to discover that it doesn't work. So it's better to have these results out there, but until PLOS One, nobody would publish them. So now they're highlighting that fact with this new collection. So if you want to browse through negative, null, and inconclusive results, you can go to this new collection. They refer to it as positively negative.
I love your title.
I think
PLOS None
is great.
Yeah.
That's just great.
Well, I would take issue with the fact that just because this person got it to work poorly and didn't get it to work at all, that you could just dismiss it and move on to something else. Because I know good examples of someone getting a good result and everyone else trying to duplicate it and getting a bad result. And then they were invited to this person's lab in utter frustration of not being able to convince them otherwise, and they sat down at the bench and did it their own way that they had in their lab back where they were from, and it finally worked. So there was some difference between the reagents.
Absolutely.
So this doesn't dismiss any of that stuff.
That doesn't dismiss any of that, but it does. If you're setting up to do something, you can look at previous work on it and say, these people had trouble with this.
Do they categorize these so I would know where to go to look to begin with?
Well, you can certainly search it. You can search through PLOS One on any topic, and you'll get some of these.
I see.
You can also, I think you may be able to to search this collection specifically. I'm sure there are ways to narrow it down. They're very, very internet savvy.
I think it would be a good example of showing people how difficult science really is to conduct, because you do get a lot of stuff that you know, but it isn't worth publishing, that still leads you to a correct answer, doing it again in another way. So it's the process of doing science, perhaps, that this illustrates best.
Sure.
One paragraph, for example, they say a selected example focuses on the lack of a significant effect of postpartum psychological distress on mothers in rural Bangladesh, despite differing positive findings in India. So if there's already been something that's been shown and you find something that shows a lack of effect, you might have trouble publishing that.
And that actually is a very serious problem, because then the whole literature becomes skewed.
Yeah. Yeah.
Like, there's this effect that exists, and it turns out that it only exists because people only published when it existed, and in fact, it may not really be real.
Or it may be country specific, or whatever.
Maybe for something like psychology, yes, it may be country specific, or it may be, there may be other factors involved.
So I've said this before, but Elvin Cabot used to be on our staff here. He was a very famous immunologist and immunochemist, and he had a famous journal that he would put all those kinds of articles in called Acta Retracta. Because if your results aren't valid and someone else didn't validate them, then you could say, oh, geez, I'm sorry, I must have done something wrong. So, I like Acta Retracta.
Well, it'll be interesting to see how this works out.
Yep.
Yes.
I think it's interesting because about 12 years ago, I submitted a paper about negative results to PLOS pathogens.
Oh. Are you in there somewhere?
No. Because they didn't publish it. The editor at the time said, we don't publish negative results.
Well, that was before PLOS 1, wasn't it?
That was then. This is now.
That was very frustrating. The guy was just, he was an editor, okay? Nowadays, PLOS decisions are made by scientists, which is good.
Is that the pre-electronic era also?
Yeah, I think so. Anyway, we published it in the Journal of Clinical Investigation, which is none too shabby. And it is a negative result, but it was important at the time. Rich Condit, what do you have for us?
I'm back in space. This is a short video.
No autoplay, sorry.
It's a short video of a time-lapse animation of the assembly of the International Space Station.
Oh, wow.
Cool.
It's actually got little captions as you go along. It tells you what the date is and what time. And there are several of these out there. I like this one in particular because the music that goes along with it is kind of a zippy jazz saxophone that I think is really nice. So I just thought it was entertaining.
Well, Rich, would you like to spend a few days up there?
Oh, yeah.
Oh, God, I would go in a minute. I would go.
Oh, yeah. Space Station would be awesome.
I would definitely go in a minute.
Have you seen Gravity?
Absolutely.
Yes.
Okay, you still want to go,
Several times.
Oh, yeah.
You're going to get blown up, man.
Oh, no, it's okay. Come on.
Silly.
All right. Very good. My pick is a lecture by Tony Fauci.
Uh-huh.
Yeah, he gave a talk on Ebola at the recent ASM Biodefense meeting.
Did you go to that meeting?
I did, but he spoke at 7:30 in the morning.
Oof.
Well,
Don't tell me you missed his talk.
I was just getting on the train.
Okay.
I missed it, but this is good. It's a good talk. Plus, his accent is amazing.
Yeah.
True.
You got to just listen to it for his accent.
He's a Brooklyn guy.
And he points
He is
out at the
Brooklyn
end, this is gives me yet. I actually listened to the whole speech this morning. He points out at the end, this gives us a segue to the Ebola SITREP, that, as we've said in the last couple of weeks, despite the fact that we're down near zero, there's still a few cases coming up, and you cannot let that go.
No. You have to
He quotes Yogi Berra in the speech. He says, it ain't over till it's over.
That's it.
Yep. All right, we have a listener pick from our friend Judy in San Diego. She sends a link to a TED Talk by Romina Libster, and it's all about the power of herd immunity. So we are assaulting the value of, we are promoting the value of immunization, and this is just another one. It's pretty good, so check that out. 69 degrees Fahrenheit, clear skies, 70% relative humidity, calm winds, barometer 30.09. Just another day in paradise in San Diego.
Right. Boring.
That really hurts.
Yeah, but you don't have any water, Judy.
That's That's a problem.
Isn't there that earthquake kind of stuff going on out there?
No,
they don't
San
do
Diego.
that there.
Don't worry
No,
about
not
it.
there.
No, San Diego really is paradise.
They just get their surfers eaten by white sharks.
Oh.
Yes, thanks for being wonderful, real people, even the flaws.
Oof.
Putting out good science and taking on the world with passion and humor. If you come to San Diego, let me know. I'd like to take you out for Mexican food. It's the least I can do.
Cool.
I wouldn't.
I'm on my way.
Wouldn't turn that down.
Offers also for Elio, even if he lives here. What flaws, Judy?
Well, you're on the 13th flaw, and I'm on the first flaw of my building.
TWiV 328 and all the other TWiVs are at iTunes and TWiV.tv. And do send us your questions and comments. We enjoy talking about them. TWiV at TWiV.tv. Thank you, Dickson Despommier here at Columbia University and verticalfarm.com.
It's a pleasure.
Any good plans this weekend? You're going to Miami.
I'm going to Miami. Yeah, my wife and I are going down to witness the wedding of her cousin.
Very good, Dickson. Don't cry. Don't cry, okay?
I'm not going to cry. I'm not going to cry. They're one of the first people to get married under the new same-sex couples.
Is that Proposition 8?
I doubt it.
No, not in Florida.
No, it's something else. But it's a celebration of diversity and of freedom of gathering.
When are you coming back?
Monday morning.
All right, enjoy.
We will.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. And I want to wish all of you a happy Pi Day tomorrow. And all of our listeners, by the time they hear this, will be belated Pi Day. But tomorrow, 3.1415,
cool.
and especially at 9:26:53, morning and afternoon.
Nice.
Excellent. Thank you, Kathy.
I heard the bakeries are going crazy with this idea.
Oh, yeah.
Hey, have a pie.
Baking a pie, have a pie at 9 in the morning, 9:26 in the evening.
Nice.
Yeah. And my brother pointed out that those people that put the day before the month, Europeans and people in South America and so forth, they never get to have a Pi Day.
Oh,
Sad,
bummer.
very sad.
There's no 14.
They don't know what they're missing.
Yeah.
Rich Condit is at the University of Florida in Gainesville. Thank you, Rich.
Sure thing. Great time.
83 degrees, huh?
Yep. That's still right. Yeah, it's still 83 degrees. That's right.
Wow, amazing.
This is the time of year. I mean, that's like a 40-degree Fahrenheit difference between us and you, even more. It's amazing.
Yeah, but then it gets too hot, right?
Well, depends on your perspective. I'm okay with the hot weather.
Alan Dove is at turbidplaque.com. You can also find him on Twitter. Thank you, Alan.
Always a pleasure.
Where you are Alan Dove on Twitter, right?
Yes.
Very good. I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.